Thank you, Ben. I couldn't have had a better introduction or setup for my presentation. So, certainly, we're going to talk about all of those aspects related to targeted drug delivery. And Ben is absolutely right in that it is probably the biggest topic that comes up when we're engaging with customers for nasal administration. So, to kind of dig into this a little bit, we're going to talk a little bit about the nasal physiology and how that presents some challenges to think about for the drug delivery. And we're going to highlight that with a few examples that you see here. Some that you've probably heard about, nasal vaccines and drug delivery. But we're also going to cover a few novel receptors that you may not have heard of. Um, there's a device aspect as well as a formulation aspect to consider. And since this is a growing area, I wanted to wrap up with a few tools that we could talk about to help understand the development as formulators. So, today I think intranasal administration is quite exciting, and that we're not really thinking about local indications. While certainly allergic rhinitis and polyps are troublesome to people, but we're using the nasal cavity now for rescue medications such as for seizures or opioid overdose. We're utilizing it to treat dry eye disease and depression, and certainly a good example of targeted delivery is the recent approval of the Impel platform for migraines. Um, so certainly, there's a lot of opportunities, exciting for delivery and nasal administration. And who knows, tomorrow we may see treatments which are already in development for unmet needs such as Alzheimer's and MS, and continuing to see advancements for treating depression and anxiety. So I would be remiss if not talking about the nasal cavity and where these targets are. So I want you guys to start thinking beyond treating allergies, where the nasal cavity may have as many as really three distinct areas that we may want to target depending on our indication. The first one I'm going to talk about is CNS delivery. That's a very hot topic. And you can see in the side view of the nasal cavity there on the left hand side, there are nerves that innervate from the olfactory bulb into the nasal cavity. And those are potential targets for transport into the CSF and to the brain. There is a demonstrated pathway of CSF transport into the deep brain. But there are some challenges that we'll talk about in a few slides. Another area that is of interest is that red circular area. That's the NALT, or the nasal associated lymphoid tissue. That's a potential target for nasal vaccines. And certainly in today's day and age, vaccination is a hot topic. And finally, if you're thinking about systemic drug delivery, you want to maximize deposition in the turbinates, which are those yellow projections there, where the turbinates serve a purpose to increase the surface area of the nose, and they make an ideal target for systemic drug delivery. So we're starting to think about the nose in these distinct areas. But to think about how we're going to get the drug there, because that's the first thing, right? We want to have deposition. We need to understand, what we're up against. And the nasal cavity is, while it has a large surface area, it's a very narrow and convoluted passageway. Shown here on the right-hand side are some flow air streamlines that are representative of 4 different adult volunteers, and they're color coded with the red being the highest velocity of 400 centimeters per meter or per second. Sorry. And you can see in these streamlines that the area where there's the greatest airflow is right at that nostril, the front of the nose, because there's a bend and a narrowing in the airway. So that really does lead to deposition in the front of the nose, or along the floor of the nasal cavity. So we need to take these physiological considerations in mind when we're developing a drug for a targeted area. You'll also note in these images that even though there's only 4 volunteers here, you see a tremendous amount of inter-subject variability between where the airflow, generally moves through an individual's nasal cavity. The other aspect that we need to also consider is mucocele clearance. So if we're inhaling a powder or an aqueous nasal spray, Those particles are going to deposit on the mucus layer, which happens to be a bilayer. There's a gel layer and then an aqueous or more aqueous layer underneath. That's where the cilia beat and move the mucus to the back of the nasal cavity to be swallowed. You can see in the cartoon diagram there on the bottom right, the blue arrows indicate the direction of mucus flow. And it's actually a very rapid process. The nasal cavity an adult human that's healthy can clear the nasal cavity in about 15 to 30 minutes. So, in some cases, for these unique therapies, we may have a window in which we need the drug to either be absorbed or interact with the cell or the target cell. The other thing that I'll point out is that the density of cilia is greater along the lower regions of the nasal cavity. There is less cilia up in the olfactory region, which is the top of the nasal cavity. A few other things to think about from targeted delivery is that while the mucus is primarily aqueous, it's 95% aqueous, there are also people that are considering using mucin to their advantage to improve retention and possibly using excipients that may bind to the mucin. So that's also becoming attractive in the area of targeted delivery. So, we've learned about some of the challenges. We're going to switch now to talking about a few examples. And I wanted to lead with vaccination, intranasal vaccination. Certainly, it is a hot topic given the era that we're living in. Um, and when we're talking about intranasal vaccination, the target is the NALT, which is in the back of the nasal cavity, as well as the dendritic cells that line the nasal epithelium. Um, when considering giving a drug nasally, we start to talk about aspects such as dose volume and retention in the nasal cavity. But some of you in the audience may be wondering, why would we use the nose for intranasal vaccination? Um, One of the advantages is that it induces mucosal immunity. And I think this cartoon drawing that I've modified from a publication shows it really well, in that intranasal administration of vaccines triggers an IgA response, which is that mucosal immunity. It also can trigger a systemic immunity, which is the IgG response, which we normally would get through an intramuscular vaccine. But another key advantage of the mucosal vaccination is that it ramps up those B cells and T cells, which actually help to kill the virus and prevent viral shedding. So there's another really important advantage for intranasal vaccination. And as a result, we see an uptake in these programs. There are 145 nasal vaccines that are actually in clinical trials or development. And interestingly, only a third of them are actually COVID related. So there's opportunities in infectious disease as well as therapeutic vaccines, which for things like head and neck cancer. Along the infectious disease standpoint, the nasal cavity is also being considered for prophylaxis of this against the SARS-CoV-2 virus. We heard earlier this week in the presentations about where the virus deposits and sort of takes hold in the nose and that mechanism. So that makes sense that we want to deliver a treatment to where the virus first settles and takes hold. And this is an example of a nasal IgG formulation that's being developed as a prophylaxis for this infectious disease. In this case, we're actually targeting the front of the nose and the turbinates because we want to target where the virus deposits in the nose. This is actually a poster presentation here at this DDL conference, so you can go and check it out to learn a little bit more about it. But I thought it was interesting because, IgG is a very large protein. We're starting to see these types of, nasal programs being developed. And this is a good example that you're able to actually formulate a IgG protein formulation. Um, the studies so far have shown that after you spray it, the formulation, we're not getting any aggregation. It's showing some stability, as shown by the SEC chromatogram. As well, we're able to spray the formulation, the plume geometry images there on the bottom. The plume angles for the two concentrations of the formulation are about 41 to 42 degrees, and the droplet size is around 50 microns, which is a pretty good indicator that it's going to deposit in the target areas of the nasal cavity. Also, we're going to talk about another hot topic, which is CNS drug delivery. Um, so if you deposit a drug in the nasal cavity, it could be absorbed into the systemic circulation and potentially cross the blood-brain barrier through the blood pathways. However, as this schematic shows, the blood-brain barrier, which is the image there on the right-hand side, has a special layer of endothelial cells that act as a really strong barrier to prevent transport of molecules into the brain. It's really acting as a defense mechanism and to maintain homeostasis in the brain. Um, it's generally limited to small molecules, particularly those that are less than 400 Daltons. So it's not a slam dunk that you're going to be able to deliver, across the brain barrier through intranasal or through systemic absorption. So that leads people to start thinking about nose to brain delivery. There we go. It advanced. It's I have a question mark because it still very much is a question. It's an evolving area. So the target here is the nerves that innervate the nasal cavity when we start talking about nose to brain. And this is a recent example that was published by Dr. Susan Kraft and her colleagues at Wake Forest. This was a study that looked at adults, or 49 adults, who received either intranasal insulin or a placebo for a year. And in this study, they were looking for a biomarker that is indicative of Alzheimer's progression. So that biomarker is white matter hyperintensity volume. And that's what we see graphed here on in this figure. We have the biomarker versus various regions in the brain. The gray is the insulin and the white is the placebo. So having less of this um, biomarker is actually a good indication of improvements in cognition. So this is demonstrating that somehow insulin in these volunteers is making a difference. It's getting into the brain, um, and we know it's not going systemic because insulin does not is not significantly absorbed um, from the nasal cavity. So this is some indication that the nose to brain pathway exists. And the theory for insulin is that there are specific insulin receptors that are both present in the brain as well as those olfactory neurons. Now we're going to talk a little bit about some unique aspects of targeted delivery utilizing some other features of the nasal cavity. This is an example of a drug that's in development with a company called Vistagen, where they have a novel class of pherine molecules that are used for anxiety. And what's interesting is that this drug triggers a series of nerve transduction to cause that anti-anxiety effect through the limbic system. So the drug deposits, or the target is the chemoreceptor that's in the vomer region of the nasal cavity. It's that little black circle in the nasal image that's located towards the nasal septum. So the drug, which is PH948, deposits in that region and starts this cascade to have the anti-anxiety effect. And the functional MRI images there on the right-hand side, where the dark orange, represents demonstrating that the drug is actually reaching, or there's actually a signal. I shouldn't say the drug is not actually entering the brain, but there's a signal process that's causing that response. Another example of a unique pathway is the utilizing the trigeminal parasympathetic pathway for dry eye disease. This drug was actually just recently approved by FDA. It's varenicline, which is used to help with tear film production and homeostasis to treat dry eye. I chose this example because in their package insert, you can see there on the right hand side, they're actually having you direct the nasal cavity, or sorry, the nasal spray away from the septum and aiming towards the ear to target those particular nerve pathways. So you can, as you can see from these examples, that I think the nasal cavity, we're getting very hyper focused on specific sensors and nerve pathways as a means to target specific molecules. And, we've seen a couple examples where the nasal spray was oriented in specific directions. And some things that we can use, some tools that we can use to understand deposition are nasal casts. This is an example of an Aptar VP7 pump with a modified actuator, specifically designed to try to target the olfactory region. The device in this case, the two examples here are where the depth of insertion in the nasal cavity was changed. So the blue is at 5 millimeters in insertion depth, and green is 10 millimeters insertion depth. And you can see if you look at the olfactory bar there, the percent deposition can change as much as 30%, depending on how deep that device is inserted into the nasal cavity. Administration angle can also matter. So this diagram shows two aqueous devices, an Aptar VP7 and an Aptar UDS device that's actually filled with liquid. The x-axis shows different angles of administration that's used in this nasal cast model. So you can see with the device on the right, it's fairly agnostic to the administration angle. We get roughly the same amount of deposition in the olfactory region. But with the device on the left, depending on how the device is used, we can actually increase deposition in the olfactory region. So this does to me indicate that there is a device aspect when it comes to at least when it comes to targeting the olfactory region. But as time goes on and we get more and more involved in different programs, as well as, surveying the literature, I personally am convinced that the device is only part of the solution, and that we also really need to understand the formulation considerations to improve transport or prolong retention. Particularly with the larger molecules, and the IgG example that I talked about earlier is really, indicative that, we have the tried and true excipients that be used to increase viscosity and prolong retention. But we're starting to see a growing interest in the use of mucoadhesion, to prolong retention for vaccines, so they have time to interact with the immunocompetent cells, um, as well as perhaps some penetration enhancers. We have a few products now on the market that use penetration enhancers, the sumatriptan and the glucagon nasal powder. But what is particularly interesting to me, and I think this is an evolving area, is the use of nanoparticulate systems to deliver the payload. And I think we need a lot more understanding here. Like, people will ask me, well, what size nanoparticles are optimal for delivery to the nose? And I think the answer is really, we don't quite know yet. So it's an evolving area, but it has a distinct interest in CNS delivery. And it also triggers some concerns in that you do need to use a biodegradable type of system if you're thinking about nanoparticles in the brain, because the brain could be sensitive to those particles. You could have CNS toxicity. So I think this is an area, for me, the area to watch. And it's, I think, a really interesting time to be involved in targeted nasal drug delivery. Finally, I want to conclude with a few thoughts on how we're going to quantify this as formulators, and in terms of, figuring out what we want to take into the clinic. The first three bullets there in gray are the tried and true things that we know about: gamma scintigraphy imaging. The spray characteristics, droplet size, and plume geometry can be indicators of where the product would deposit in the nose. And you've already seen some examples of nasal casts. Though one of the challenges of nasal casts is that they're static. We don't have the ability to really simulate the mucocele clearance yet to understand the retention piece. There are people that are working to advance this area in terms of utilizing nasal casts to have them be a bit more functional, as well as linking them to CFD modeling, which can be used to, from a predictive PKPB standpoint. What I've seen other folks looking at are mucus secreting nasal epithelial cell lines to try to understand penetration, and then some growing area in terms of means to quantify that interaction with mucin, if that's your target for mucoadhesion and adhesion. Some equipment is listed here. I think one of the challenges that um, there's not really a standard way for any of these models, in a given laboratory. So it's hard to compare one work research to another. But it's a growing area in which I think people are, utilizing these techniques to truly understand what's going on at a more sophisticated level in the nasal cavity. So with that, I'll wrap up and I'll thank you for your time. I really do see just from the conversations we have with customers that there is the use of targeted when we talk about nasal deposition is a key term these days. I hope you've shown that there are some device considerations, and certainly one device may not fit all situations, and that if you're targeting the olfactory region versus the nault, it may be a different device platform. um, how the person is using the device is certainly going to make a difference, particularly in the nose to brain or CNS delivery space. But really, I think we need to start paying attention to the formulation aspect as well to really maximize the uptake for indications, particularly in the CNS space. Um, Thank you, everyone, for your attention. It's a pleasure to present at DDL. I wish I was there in person. So hopefully we'll see everybody next year, and I'll be happy to take some questions.
We wish you were here in person too, Julie.
Yeah, thanks, Ben. Yeah,
We've got a lot of questions coming through, and we'll try and relay them to you as best we can.
that's good. I'm working on my second cup of coffee. It's 7:30 or almost 8 o'clock.
Oh wow,
Yeah.
we'll try and stimulate you.
Yeah.
A few questions around the nose to brain
Sure.
delivery, and the observation that the examples that you showed and some of the other ones out there tend to rely on looking for biological responses in the brain, and there's that question of quite how much drug gets to the brain. So of the administered dose to the nasal cavity, what evidence do we have in terms of the actual amount of drug that reaches the brain to be causing those effects? And
Yeah.
the supplementary is kind of what experimental setups do we have to try and probe that a little bit more?
Yeah. I mean, it's a challenge. I think right now we're looking at bioavailability in the brain is still less than 0.1%. So, the molecules that I think that are being successful are potent molecules. I think that's where like nanoparticles or other particulate systems come into play to try and increase the payload that can actually make it into the CSF for the brain. It's quite tricky. If you're looking at an animal model, the only way that you're going to be able to determine if there's uptake in beyond the CSF, we can assay the CSF in an animal fairly readily. But to really know if the drug is making it deeper into the brain, you need to sacrifice the animal. So those experiments become challenging and more expensive. And then it's, as you can imagine, in the human, it's even more challenging because you can sample the CSF, but you're obviously not going to be sampling brain tissue in a human. So I know the example that I shared with you from Dr. Kraft. I know they have a study planned they're going to tag insulin with some sort of marker, um, and to follow it, and see if they can understand the exact pathway. that it's making the insulin is making it into the brain. Right now, it's a theory that it's targeting those receptors for transport. So I think it's good old fashioned targeting or tagging a molecule and trying to follow it through imaging studies, um, or utilizing a biomarker at this point.
Yeah, it'd be fascinating if that could be linked to device and formulation factors as well.
Yeah, so I didn't talk about it here, but there's also a paper that's out there. Dr. Kraft has done some work in that space, particularly with insulin, where she had looked at two different devices. Um, and one of the platforms did produce results in the CSF and another didn't. So there does, at least in that example, there was a device related aspect for sure. So she had to switch back. She was, anyway, you could read the paper, but she had to implement additional protocols to select a device for her insulin studies.
Yeah, great. Okay, I think Alex would like to ask a question.
Yes, I was going to ask a question that one of our delegates has asked, and it's to do with the intranasal vaccination formulation approaches that you mentioned. And the question is, will you have to include potent adjuvants in order to do that? And then connected to that, are there similar tolerance mechanisms in the NALT that stop unnecessary immune response to environmental antigens?
Interesting. So there are 4 intranasal vaccines that are been approved over the years, and they've all been influenza. They've been live attenuated vaccines. There are those that say a live vaccine intranasally is the best way to go, but I can tell you most of the ones in development are not. They're mRNA based or other some sort of other particulate system. And one of the challenges in nasal administration is the selection of an adjuvant. So the nanoparticle or chitosan can also help, potentiation or, uptake into the immunocompetent cells. Chitosan, so in this case, can, serve two roles: to increase retention and also act as an adjuvant. But one of the challenges in nasal vaccination is we don't have this wide list of adjuvants that we can pick from that are known to be useful and safe. So depending on what you're choosing, you could be opening up the door to look at other tox considerations as well. So certainly, if it's not a live vaccine, you're going to need an adjuvant in your formulation. I don't know the answer to the tolerance question, to be quite honest. It's a good one. I think it's conceivable, but I don't know enough to be dangerous with that question.
Great, thank you very much. I think Michelle downstairs has got a question for you, so if we can cut to her, perhaps.
Hi, thanks, Julie. Thanks for a really interesting talk. And it's just sort of, it's so exciting to hear that, people are really looking at nasal delivery for totally different modes of action, a bit like lung, sort of, different types of delivery, particularly around CNS delivery. And I guess my question was linked to probably one that came through from a delegate as well, who was asking about challenges in ensuring consistent doses to patients on a day-to-day basis. And mine sort of stemmed from a little bit around that you mentioned, that there's potentially opportunity here for nasal delivery for things like Alzheimer's. There was had a talk yesterday on, I think, nasal delivery of ibuprofen, I believe. And I guess as having family members who have reduced cognitive function, I'd be curious to understand your views of what you would see as the challenges of what we would need to do to optimize delivery to those types of patients, to almost take them out of the equation to get the doses that they need to be effective.
Yeah, that's a good question. And I myself get a little bit nervous, quite honestly, like the one slide that I showed where, you can change deposition by just changing the depth of insertion by 5 millimeters. I mean, you put that in the hands of the patient and, you tell them to, you almost are going to need to have some sort of specific device to the design so they always orient it in the right, the right spot, whether that's some some sort of guide that's associated with the device, because certainly in the hands of patients, whether or not they're going to be able to follow the instructions. So I think there is a design aspect to ensure that it's reproducible. In terms of cognitive, function, this is perhaps where we can rely a little bit on what we've learned for some of the drugs for emergency use. We've known that, caregivers can administer, the benzodiazepines for seizures, or we have the Narcan. So, we know that those devices have a history of being amenable to a caregiver, delivering a dose. Perhaps in the world where there's a cognitive issue, this is where, digital connected devices play a role, where there's some sort of reminder. Did you take your dose? Of those sorts of things. So that might be something that gets as an added on layer to the device aspect in that patient population.
Julie, thank you very much. Lots of questions are coming through, but I think really in the interest of time now, we need to move on. So thank you very much.
Sure.
Thanks for connecting.
Yep, thank you. Certainly, folks, feel free to email me. I'll be happy to try and answer questions. So thank you, Omar, and everyone.
Super. Thank you.